Okay, so the next speaker is me, but it wasn't supposed to be me. This is the title. I'm sure you've all seen that. What's happened was I had myself an MSc student this year, Sonia Lee. So this is just a nice picture of Sonia. So I was hoping that Sonia would be here talking about the work that she's done for me this year. But unfortunately, Sonia is on a well-earned holiday in Japan. So you have me instead of Sonia. So what prompted me to look at gall midges? Why would you? So basically, I started running a Malaise trap at a local nature reserve near me. I should say near me, and I was doing it because the people running the nature reserve have got permission to rewild the nature reserve, and I thought it would be interesting to have good solid data on the Diptera before and after that process, which obviously there's lots to be said about rewilding and what it actually is and whether it's effective or not. Let's not go there. But I thought it would be interesting to look at before and after and to see what the impact is on the flies. So I've done my before flies. I'm not sure that there's much happened on the rewilding front. There's talk of cattle, and I think there have been volunteer days for looking after cattle and that kind of thing, because it is right on the edge of London. So I think there's a good chance your cattle could get nicked. I don't know. Lads on mopeds driving by. So anyway, so I'm looking through my Malaise trap, and I'm sure anyone who spent a lot of time looking through a Malaise trap has had the same thoughts. Gosh, another cecid. How many can there be? So So while I was hunched over my Malaise trap sample, I came across a paper by Rudolf Mayer's group. So this one down here, oh, what do I want? I'll have a laser pointer. Anyway. So this paper here, Convergence of Dominance and Neglect in Flying Insects. So basically they put up 39 Malaise traps. They covered 5 continents, so they missed out Antarctica and Australia. And this figure here taken from the paper, this top line here is the cecids. So on average, 20% of what they were getting in the Malaise trap from everywhere was a cecid, a gall midge. I mean, there's a big standard deviation here, 13. So we're talking between 5 and 30% of your total catch is gall midges. And I don't know about other people, but at the moment, all the gall midges from my Malaise trap have just gone in a tube and they're on the shelf. So I just was thinking, well, this is crazy. This is such a big proportion of what's out there. We need to start understanding it. So this was all in my head. And yes, I think maybe not. If you actually add up these 3 in the green boxes, so this is the cecids, the platygastrids, and the eulophids, then 25%, a quarter of all the insects flying around everywhere all of the time are either a gall midge or a tiny wasp looking for a gall midge. So that's an insane statistic. Can this really be the case? So what do the experts say? Because there are experts on cecids. So these are some books. So Raymond Gagne, I think he's in Canada, and Matthias Yashov. I mean, there are other. There's a group of them. Sadly, none in the UK. I think since Keith Harris passed away. But Gagne and Yashov are behind the World Catalogue, which is available online. So what do these guys say about how many cecids there are in the world? So, well, they're pretty coy, really. So at the top, we've got quantification of their global species richness is an insurmountable task. And just in the catalogue, they just quietly say, the gall midges are probably the most diverse dipteran family. So not a lot of detail from these guys. So what data have we got available to us? The big one is this GBIF, and this is, you've probably all looked at it. It's an international network and a data infrastructure, and they're taking all kinds of data there. So it's everything from an occurrence record that you might create on your smartphone to sequence data in some big DNA thing. So we've got in excess of 1.8 million Cecidomyiidae records, and these are from 7,060 species. So I think that's our sort of global total for described species of cecids. BOLD is the big DNA biodiversity database, and that one for insects anyway is mostly or almost exclusively mitochondrial CO1 barcode sequences, and they've got in excess of 1.3 million records, but only 12,600 of those 1.3 million records have a name attached to them. So that's less than 0.01% of your records. And they're from just 916 species out of the 7,000. So there are people out there generating a lot of DNA data, but very little of that data for the cecids has got a name attached to it. So it's going to be difficult to use the DNA to identify all the cecids in your Malaise trap sample at the moment, with the current situation. And they talk about, BOLD talks about bins, and they've got a complicated algorithm, and I believe mildly controversial, because not all of it is that all of the information on how the bins clustering tool works is publicly available. So it's a slight black box. But what it does is it clusters the DNA sequences on similarity, and it comes up with these OTUs, which are operational taxonomic units. So think of those as putative species. So the DNA would suggest that there are in excess of 96,000 cecid species. So that's for the stuff that we've got at the moment, the current DNA yield. Okay, so are there any papers from nearer home? So these are some papers that I found. These are all quite recent. A lovely paper, June this year, from the Norwegian Journal of Entomology, team there. Basically, they collected plant material on a giant scale from across Norway, and they reared the insects out of the obviously galled bits and pieces and things with leaf mines. And from doing that alone, so this is excluding quite a lot of the cecids that aren't gallers or leaf miners. They added 93 species to the Norwegian list with just one study. But the list was quite short. It was only 240, which is probably a wild underestimate of how much there is in Norway. And then this study here from Canada, this is the oldest one, this is 2016. And this is Hibert and co. So these are the people behind the Barcode of Life project. And this was a DNA-based study, and they did some clustering. And based on their clustering, they're saying there's in excess of 16,000 cecid taxa, so these operational taxonomic units in Canada. And the previous list was 1,600, so that's a 10-fold increase on the number of taxa, if their number crunching is correct. And then this study at the bottom from Germany, previous list 849, did a bunch of DNA, some numbers, and they gave us a range. But if we take the average of that, we're talking about a 5-fold increase in the number of taxa over, the previous list. So what does this, I've got some, these are fun, aren't they? Okay, so what would this mean for the UK? So at the moment, we've got a very well curated list of the flies that we think are in the UK. This number's from the April this year version. So 658 is your total for the cecids from 154 genera. So if we took the German 5-fold estimate, that would be 3,290. But if we went for the upper Canadian estimate, we're talking about 6,580 species of cecids in the UK. So and available data for UK cecids in these databases. We've got over 17,000 in GBIF. The NBN Atlas has got nearly 18,000, but from only 164 species. So a lot of the UK things that we know or in the UK are just not, they don't appear in the Atlas because people aren't recording them. And BOLD has just 297 sequences for the cecids, but I know that that's not all the sequences that are available for the UK. There are really big data sets in the pipe. It's just that they haven't made it to the end of the pipe, and so they're not publicly available on BOLD yet. So that number, I think, is a big underestimate of what's going to be available shortly. So I then came across this paper from Emily Hartop, and what they're proposing is a sort of alternative to trying to do the taxonomy on these mega diverse groups like the cecids, where only a small proportion of what we think the real fauna is has already been described. So how can we do this in an efficient fashion so that we get it done before they've all gone extinct, because we've wrecked the planet. So their proposal is that you have two types of data. So your first type of data is your cheap, easy available, easy to get data, and you do that for every specimen. And then you go back and you do some complicated stuff. You'll see it in the next slide, which has a complicated figure. But your second data, which is your more difficult to obtain, expensive, time-consuming data, you only do that for a few individuals. And this is where you're saving on the effort and the amount of time it takes you to describe your fauna. So they come up with this workflow for achieving this in a kind of, sensible way that means you're not missing information, you're not missing diversity. So they did this for this paper up here. The 22 paper is for phorids. And I think it was Mark, was it the Swedish phorids? Okay, so using the material from the Swedish Malaise Trap project, and they ended up doing the morphology because they did a lot of extra work to validate what they were doing. But if they had just used their workflow, they would have looked at 5% of the flies to do the morphology, and that works out at just 2.5 specimens per species. So that sounds like super efficient process. So my thinking was, well, can I do this with the cecids in the UK? So that was what poor Sonia was up to. We were doing little pieces of work to see if the components of this workflow would work with a different group, i.e. the cecids in the UK. So step one, you acquire your easy, cheap data, and they're basically saying that it's now so easy and cheap to get a DNA barcode that that's your easy, accessible data. So you do that for every specimen. And then step two, they did lots of validation work, and they got the DNA for everything. They put it into these OTUs, these bins, these clusters, and then they collected a load of statistics on the OTUs and the bins and clusters. And they gathered up all this data on them and what they're like. And then they went back, and the validation step was to do the morphology for everything as well. And then they compared the morphospecies with the bins. And then they found the ones that were in conflict, where your bin didn't agree with your morphospecies. You were having there, and they found that they didn't have any cases of a bin splitting across. No, there were bins that contained more than one morphospecies, but there were no morphospecies that were split across two bins. Yeah, so morphospecies were within bins. Is what I'm trying to say. So they looked at it, and the conflict ones, they looked at the statistics that they'd gathered on these bins, and they did find some statistics that were higher if your bin was going to be in conflict. So they were then able to use those as tools to examine all of the bins and pick out the ones that were likely to be in conflict. And so if your bin was not going to be in conflict, if the statistics were such that it was going to be, the experiment had shown that it would agree with the morphology, you just looked at two individuals. So the most distant haplotypes within your DNA barcodes, and that was it. You're done. You've confirmed. If they're the same, you're happy. That's one of these, a confirmed cluster. So you just look at 2 individuals and away you go. If you're going to have conflict in your bin, then you go in and you have to look at an individual from with every different barcode haplotype. And so you've got to do more morphology in your potential conflict bins. So that's how the process is supposed to work, and that's how you're saving on time and effort. What did we do? Oh, okay. So this is what we got out of Sonia's MSc project. So the Cecid people, they're using Canada balsam thinned with xylene, and they're using beechwood creosote to clear the specimens. And I looked at Sonia, and you've seen the picture of her, delightful young girl, and I thought, I don't think I should expose her to xylene and beechwood creosote. So I persuaded her we needed to come up with a protocol that used Uparal, because that's not thinned with xylene, and we used clove oil to do the clearing. And we produced some okay slides. We were able to do some morphology. They were good enough. But there are people, I think there's a lady here works on Hemiptera, Diana, and she uses Canada balsam thinned with histoclear. I don't know if anyone's used that. But the kosh on that looked a lot less scary. So maybe that would be an alternative to the Uparal. So that's something I might explore in future. Maceration in KOH, we needed to do it for some specimens, but not all of them. Some of them, once they'd hung around in the Malaise trap sample, they were really quite ghostly and clear already, and they could go straight on a slide. Too long in ethanol, and the specimens were becoming brittle, and it was difficult to make a slide preparation. So I don't think you're going to be able to do morphology on Cecids from like really ancient Malaise trap samples. I don't think that's going to be a goer. And also there was an issue with sort of high rate of damage to the specimens, because Cecids are an unfortunate combination of small and also very flimsy. So the fact that they're in a Malaise trap sample in the soup, they were losing lots of antennae, legs. And also, if they're all going for DNA extraction, that's also, that softens them, and it's lots of moving from tube to tube and being picked up with forceps. So we were losing, bits and pieces in that process as well. So I think 15% of the specimens that we produced a sequence for were so kind of mangled that it wasn't worth putting them on a slide. I mean, that's a big rate of attrition. 61% of them we couldn't put the wing on a slide. It was just too torn and mangled. 53% of them had lost their antennae, and if you look in the Cecid literature, they do look at sensory hairs and how many segments there are in the antennae. So these things are really significant issues. So this is not by any means going to be straightforward. But we were able to use morphology in some instances. DNA, we successfully got DNA out of Malaise trap samples that had been hanging around for between 6 and 14 months. And we did it with the cheapest extraction protocol, cheapest chips, and we still got decent yields. So that was encouraging. I think the 63% success rate on the sequences is more to do with the fact that Sonia was a complete novice and she had a short amount of time to do this work. So I think, you do pick up skills and your success rate improves. So I'm not sure that that's a meaningful statistic. Blast searches. So the sequences that we got, we put them into the big DNA database. It's called doing a blast search, and you get back some names. But as I said earlier, a lot of the sequences are, they're nameless, and there's only a small proportion of the names that are in there. So we got a 31% hit rate for flies from the UK. So at the moment, you can't do your Cecids based on DNA because there's just not enough names in the DNA database. So hits, we got 9 Bryomia absectra, this Coco I can't say that, and this one at the bottom, which is the raspberry cane midge. So that's no surprise. So not very many. So using morphology, some of them were pretty straightforward. So this is the one that I can't say. So this is our specimen that we've sequenced the DNA for, and this is from the diagnosis in the literature, and I was pretty happy that they were the same thing. So yeah, thumbs up. Sometimes it's nice and easy, all tickety-boo. Okay, so this is the Bryomia absectra, and we got 9 of these out of the ones that we'd sequenced. Clearly, Sonia's slides are not all beautiful, but and this is a syntype out of the collection here at the museum. And this is the genitalia details from the diagnosis in the literature. I was reasonably happy that they were the same thing, but not, I'm kind of like, I can see differences here. I'm not totally sold on this. So, and I will, moving on from there, I thought, well, I should look at the other Bryomia species, even though I only got hits for Absectra. So these are the other, so in the UK, the list has Bryomia gibbosa, Bryomia bergerothi, and Bryomia absectra. But I've added in Bryomia producta. We haven't got any in the collection, so there isn't a specimen image here. Producta was at one point sunk into, no, I think absectra was sunk into producta, and it was a junior synonym. And then they noticed this little process here, and they said, oh no, this is a good species. So it got resurrected. So it's once again, absectra is, well, I think they fished out, I can't remember how it worked, which way around it was, but these two guys were together and then they got separated again. And the list, Peter Chandler's list does say that producta could be in the UK. So we included it. And basically, I'm not sold on most of these, really. I feel, this is much more extended, and this process in the middle isn't really, reflected here. And this seems more broader and more truncated. This one, yeah, I don't feel like this is the same creature. This is the absectra, this is the syntype, and this is one of the figures. And this is producta, and supposedly we've got this little thumb, so we should be able to spot that. So I think that there's, who knows what's going on here. There's lots to be unpicked. So not straightforward. And I thought it was interesting that the thing that we got the most hits for was a problem. So we did some haplotype maps. So this is part of your workflow if you're doing the large-scale integrative taxonomy process. So each of these circles is a unique barcode sequence. And if there's lots of individuals with the same sequence, you get a big circle. And if you've only got one individual with that particular sequence, then you get a tiny circle. And the number of hashes on these arms tells you how many differences in the sequence there are between those two circles. So B is, what is B? B is producta. So this is the one that we might have in the UK, but at the moment is not on the list. And these are sequences we downloaded from the DNA database. We didn't generate all of these ourselves. So what does BOLD say about these two in terms of bins, these operational taxonomic units? It's giving us 2 for Bryomia producta. So I suspect that there's a cut here where we've got these long arms with lots of differences down here. So I suspect these guys are all good, tickety-boo. We're happy that this is a taxa, but we think that these guys are just too far away and this could be something else. This one is gibbosa. We only had 24 sequences, so that's quite a small haplotype network. So the one that we did sequence was absectra. This is the haplotype map for absectra. And basically, this is not one taxa. BOLD says there are 10 bins in here. So potentially 10 different species that are all at the moment going under the same name. So, And interestingly, our sequences, it's not just the ones that Sonia generated, there were ones already in the database, but ours were all in this arm over here. And this arm is completely orange. So these guys are only from the UK. They haven't found any of these haplotypes anywhere else in Europe or the rest of the world. But there are sequences from the UK, you know, throughout this big web. They're all over the place. So it's not that, you know, this is what's going on in Canada. Canada is where Boulder based. So all of these, you get a lot of sequences from Canada because that's where the project is based. So they kind of swamp your maps. But so, yeah, so basically not at all straightforward. And if I use the workflow, I'm supposed to look at an individual from every unique haplotype. So I'm looking at a lot of individuals, and my workflow isn't saving me a great deal of time. Yeah, so what next? Okay, so for the future. So this is the raspberry cane midge that we sequenced from Tolworth. No surprise. We need to figure out how to reduce specimen damage. So maybe you have to be very careful with your malaise trap, maybe only have it up for 1 day, plate your individuals up for DNA immediately before they've lost all their extremities, that kind of thing. There is a study where they just use 3 legs. So maybe you don't send the whole body for extraction and save some damage that way. So these are all things that we should try to do. The slide mounting protocol, Sonia used bright field and DIC illumination because the little structures on the antennae, they're really quite small, and things like phase contrast and DIC would really help with visualizing those. But I think there was a clash with the UPRAL. I think the refractive index on UPRAL is too, there was too much contrast. Yeah. So I think we might need to revisit that. I think my idea of UPRAL was a bit of an own goal. I shouldn't have done that. Anyway, so we need to refine that. Bioscan. And we're going to, I think Mara is hopefully still still there. She's going to say something very brief about the Bioscan project in the UK, but they have in the pipe, so not published yet, that's why they weren't on my slide, there's in excess of 130,000 Cecid specimens from the UK, because Bioscan is this big project where they've had malaise traps up across the UK. And so there's a lot of sequences in the pipe. And once those are produced, we could generate haplotype maps for all of our supposed taxa and just see how much this problem of, kind of multiple bins within a species, how much of a problem that is. Is Bryomia typical? And are the Cecids going to be really tricky? Or in fact, are there lots of confirmed clusters in the Cecids? And could we do a big segment of the fauna in a quite sort of straightforward fashion following the workflow? We don't know yet, but with a data set of that size, we can hopefully generate lots of haplotype networks and have a look at that and really explore that problem. Oh yes, and they're asking for AI projects here at the museum, and they've done various AI picture recognition projects, and the Cecid slide collection, it's syntype after syntype after syntype after syntype, presumably reared from the host plant maybe, and that's why you've got so many syntypes in the sequence. But it occurred to me that if you've got a big syntype run, that might be a nice training set for one of these AI recognition systems. So maybe the Cecids can go forward for that kind of thing as well. Right, and that is it from me. Thank you for your time. Does anyone have a question? Stuart.
This is very old, not my head knowledge, but I was a postdoc at Newcastle 40 years ago for about 10 years. And the seminal work on British Cecidomyiidae is called Midges of Economic Importance by Bagnall Harrison. Eslop Harrison was the professor of zoology at Durham.
Yeah.
And he slide mounted all his specimens.
Yeah.
The slides have gone black and opaque. And that includes a lot of type material. there were in my time a number of projects looking at trying to recover this material, but nobody actually came up with anything. So one of the big blocks on Cecidomyiidae in Britain has been that essentially the type specimens are lost. I mean, they're sitting there at Durham University in completely black opaque slide.
Do you know what the mountain was?
I don't think it is known. There was lots of people trying to dissolve little bits of it and trying to find ways out of it, but at least 40 years ago, nobody had succeeded. The slides did still exist.
Okay, so this is from Stuart Ball in the audience here. He's saying that there's a big collection of Cecids from the UK. Is it the Hislop? Okay, so Heslop Harrison produced a work, the Gall Midges of Economic, yeah. Okay, Gall Midges of Economic Importance. And there was a big slide collection that went with these 3-volume work, and they're at Durham University, but the slides have gone off. They're all black. And I can see Richard Lane's got his hand up, so I'm going to let Richard say something. Yeah. Mm. Okay, so Richard says he thinks it's Hoyer's medium that for the slides at Durham that have all decayed, and so there's a lot of type specimens in that slide collection that Stuart mentioned that are inaccessible and you can't look at them, and it's quite a big block for the UK Cecid field. And Richard thinks it's Hoyer's medium, which it's a gum chloral based mountain, and they are well known for going black and also going opaque. And yes, and also Richard made the point that both Canada balsam and Euparal are not going to be great for seeing these very fine structures on the antennae, and he's right. So I'm not sure what if anyone has a lot of experience with other slide mountains and has suggestions. Mark's got his hand up. Mark Welsh. Uh-huh. Specimen was still there. Okay. Okay. Yeah. 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 Okay, I clearly need to get in touch with someone at Durham University and find out if they still know where the slides are. I'm sure they do. Yeah. It's water soluble, so it should be possible to retrieve the specimens. What sort of state they'll be in, and how heavily dissected do you think they were? I mean, yeah, okay. Yeah. It's finding the bits afterwards is the hard bit. Yeah. Okay. All right. Victoria, are there any questions in the chat for me? No. Okay. Right. I think we are going to crack on.